We'll see. All right, so the discovery of cells. Now notice this image we have here contains a bunch of cells. Each one of these little boxes, centers, whatever, those are the rectangle-ish things are in each individual cells. Yes, you did. Exactly. Yeah, those are cells. So the little boxes are cells. Those are cells. We will eventually. Not today, but possibly during lab. I haven't figured that out yet. All right, so brief history. In the 1600s, Van Leeuwenhoek came up with the first light microscope. Very similar, it's actually similar to the one you saw in the picture when Mr. Hook was on there. It was kind of a tube that contained some lenses, which they used to view single-celled organisms in water. It's very similar to the ones we use now. We just refined it a bit. All right. Then we had Robert Hooke, who you saw briefly in the video in a caricature kind of way. He studied cork and called these shapes cells. So that in the 1600s is about the first time we ever talked about the word cells and said, this is a cell. It is the small unit that all living organisms are made out of. These cells are then made out of the biomolecules that we were talking about before. Yes?
But don't quarks make up protons and neutrons?
Quarks. So that is that. This is cork, as in the plant cork. The stuff like the thing we put in the end of wine bottles. Yeah. That's a cork, and that's made out of cork bark. Yeah. And he was looking at a piece of that bark. Oh, okay. So quarks are actually a different thing. The subatomic particle of a quark is different than a cork. Sounds exactly the same. Sorry. All right. In the top right-hand corner, you see the picture of what we see today. This is similar to what he saw. There were little cells, little divisions that looked like little rooms, and he called them cells. All right. And then we get to Dr. Schleiden and Dr. Schwann. Schleiden and Schwann. They studied many plant species, and Schleiden came up to believe that all plants were made of cells, and then Schwann added that this is similar for animals. So all plants and animals were then considered to be made of cells.
They work together, right?
Sort of. They probably were collaborating a little bit, but I'm not sure how closely they worked. Don't know much about that. They came up with what we call cell theory. Okay? This is based on the observations that these two made, primarily, and based on the observations before, but primarily these two. All right, there are several pieces to this. The first is that all living things are composed of one or more cells. So it could be one cell. You cannot have half a cell. It cannot have a third of a cell. It has to be at least one or more. It can be many cells, like we have many, many cells in us. All right? Whereas an amoeba has only one. The second part, don't know. Second is the cell is the basic unit of structure and organization of organisms. So just as we said the atom was the basic structure of an element, and elements then can be combined into mixtures and compounds. In this case, we're saying the cell is the basic unit of living organisms. And it cannot be divided into smaller organisms. It is what we use to build the larger organisms. No, elements are built into biomolecules, which are used to make the pieces that we use to make the cell. All right, so it's like the cell, the atoms are like the parts that make up the wood that we use to build a house. The house is the cell. The wood pieces are what we call organelles, and we'll get to those in a bit. That's what we're covering this week. And then the atoms are like the pieces that build the wood that we use to build the house. In a way, yeah. Yes, I'm aware of that. All right. And number 3, all cells come from pre-existing cells. So these cells do not spontaneously occur. They come from pre-existing cells. And so we have one cell. It will split into 2 cells. It's one of the things. Yes, that's one of the things they showed there. And so they divide and make new cells. They do not, you do not get all the pieces together and then the cell is created. They come out of other cells.
How does evolution make sense?
I'm not sure how they rationalize the original cell. I think that is one of the challenges. I haven't really studied that part of evolution's theory very much, so I'm not sure. I can find out for you, but it'll take some work. All right, so those are the 3 parts of cell theory. Now, several types of microscopes. Compound light microscope is the first one we talk about. It uses lenses to magnify the objects, okay? You need to know approximately how much these magnify so you get an idea. So notice 1500 is important. 1500 times is about the best we can do with light microscopes. No. Well, that's the only kind, we are going to be using, because that's the only kind we can afford. That's the only kind we have. But we have other types that are used, and we'll talk about those briefly. All right. Light microscopes?
Like the biggest one.
1500 is about the max.
No,
Oh, we'll get there. I've got that in a couple slides. Okay, so this is a picture of a light microscope. We have several of them. We used them in lab several weeks ago, a month ago, something. And we'll do some more. We'll do a fair bit more work with them as we're going forward. All right, you can have more complicated versions, but they're all based on the principle that we send light through lenses to magnify the object.
So that's what that little mirror is for?
The mirror is to direct the light, and then there are lenses in the tubes above it that then magnify things. Correct. That was what the mirror did. The mirror collected the light and sent it into your optics, into your lenses. Okay, so an electron microscope is the other kind that is widely used. These are very expensive. These are the ones that are probably the most accurate, up to 500,000 times the actual size.
So you could probably look at electrons?
You can't look at electrons directly because that's what we're using to do the looking. So you can't observe the thing we're looking with. But we're using electrons. Instead of using light, we're using electrons as the thing that we're kind of sending through the sample. In the previous example, we send light through and see our sample. Okay, and see how it changes. And see how the light changes in a light microscope. Now, if we send electrons into our sample and see how they change as they come out, that would be electron microscope. If you have to have a sample in a specific situation, there are 2 different kinds, and we'll talk about how they work in a second. So usually you have an electron source, you have your beam that comes through some optics, hits your sample, and then is focused through some more optics onto a viewing screen. Usually this viewing screen is then attached to a computer of some kind. Thank you, I tried. And the computer then processes it and gives us the data that we were looking for. Now, there are 2 types of electron microscopes. The first is a scanning. Should be another N in there. Scanning electron microscope, or SEM. SEM. This scans the surface of the object to get the 3D shape. So, example here. Thing on the top left, what does that look like? It's an ant, right? And so in that case, it took the picture of the 3-dimensional structure of the ant. Okay. In this case, we're not looking for the light to go through the object, Isaac, but for it to bounce off and see what it does. In the top right, what do you think that looks like? It's probably pollen. The one on the bottom left is also pollen. I don't know what this one is. It could be a little insect of some kind, and this one looks like an insect as well. So it allows us to see something that's very small and see the 3-dimensional structure. I shall wait when you're ready. So that's scanning electron microscope. The other kind is transmission, and this is the case where we're sending electrons through the sample. This allows us to see structures. Okay, that's being stupid. Structures inside the cells. So we can actually see the stuff inside the cell. We cannot see the atoms, but we can see the structure of the stuff inside the cell. Okay, here's the inside of one of these cells. You see there's some labels here, G, MT, MI, AF. These are associated with the structures next to them. And so this set of lines here, this is one of the structures in the cell. This is another structure. You also have these like weird flowery looking things. And so we'll be talking about what these are as we talk about organelles through the rest of the chapter. Okay, so that's transmission electron microscope. Now, types of cells. There are several types of cells. They're determined based on whether they have membrane-bound organelles or not. Now, when I say a membrane, what comes to mind?
Gooey.
Gooey, outside, some casing, right? Some outer casing that could be gooey, sure. Something that says, okay, we're going to define things and say there's a membrane around the outside edge. The first thing we're going to talk about, the first organelle we're going to talk about at length, is this membrane. In the video, it was those guys in white holding the sticks around the outside of their cell. That was the membrane, kind of stood around the outside and kept things in and kept certain things out. All right. Okay, so if it's an organelle that has a membrane around it, it is a membrane-bound organelle. Okay, if it does not have a membrane around it, then it is not a membrane-bound organelle. And we'll explain, I'll show you some examples of that in a bit. All right, an organelle is a small specialized structure with a specific function. So if it's a structure within the cell that has a specific function. There are many of them, and I'll give you a list of them shortly. All right, they often have their own membranes within them. So a membrane on the outside and the membrane's kind of part of the inside as well. But for it to be a membrane-bound organelle, you have to have a membrane around the outside. Membrane around the outside, which then contains stuff. If it does not have a membrane, then it is not. If the organelle is not bound by a membrane, then it is not membrane bound. Now, another way to look at this is these organelles are to the cell like our organs are to us. So if we were a cell, our organs would be our organelles. So we have organs, right? Our organs are to us like organelles are to a cell. So the cell doesn't really have a liver exactly, but it has something that does the processes that the liver does. As a way of taking in food and getting rid of food, it has a way of processing food, it has a way of building things. All those are parts. Carly.
Are those two types at the bottom, two types of organelles, or two types of membranes?
Types of cells. Two types of cells. Vivian. Yes, exactly. That's why we picked that name. So the two types of cells are prokaryotic and eukaryotic. You want to know those names. Memorize them. Getting there. Next slide. Prokaryotic do not contain membranes. These do not contain membrane-bound organelles. So it's like if our organs were not specific shapes. Example being like in some insects, they don't have organs exactly. They have all the pieces in there, but they're all just kind of mixed up, floating around. A juice of sorts, yeah. Not a worm, more like some of the insects. It's not like worms. Worms actually have organelles or organs. Okay, so you notice here we have a prokaryotic cell structure. There are several things you need to see on here. Cytoplasm. Cytoplasm is the juice that things float in. Okay. In prokaryotic cases, we have a wall or a kind of a barrier around the outside edge, and that keeps all the innards in and the outwards out, right? And then there are other pieces attached to it, but we'll talk about those in particular once we get to talking about the organelles in specifically. Yes, Carly.
What's the difference between a cell wall and a membrane?
We'll get to that once we're talking about organelles. This is just an overview, tell you what the types of cells are. All right, as a simplified structure, there's no nucleus. Okay, we have a nucleoid, just kind of this loose collection of nuclear material, but it does not have a nucleus. Okay, because a nucleus is membrane bound. And they're very small. They're usually somewhere between 1 to 10 micrometers, which is a hair is usually about a micrometer. So this is maybe one, maybe actually hair is probably hundreds of microns. But yeah, so these are very small. Yes, Vivian? Yes, correct. No. The nucleus is one of the organelles, right? But you can have multiple membranes within an organelle. We'll get to that. So bacteria is usually what these are. Prokaryotics are generally bacteria. Eukaryotic cells are the ones with membrane-bound organelles. This will help answer your question, Vivian. There are a bunch of organelles. We will study these near the end of the week. And they're membrane-bound. Let's say, for instance, if we look at this one here, this is called the Golgi apparatus. Or actually, let's look at this one up here too. And this one up here is called the mitochondria. Okay? In the mitochondria, what we have is we actually have a membrane around the outside edge, and we have a membrane that kind of makes a structure inside. So there's two membranes. And so this one is a organelle, the whole thing's an organelle, that contains another membrane. So this is an example of one where we have a membrane within the organelle as well as one on the outside.
On the outside of the organelle, and the cell wall is on the outside of the cell.
Correct.
Okay.
Okay, so membrane-bound organelles, right? They have a nucleus containing DNA, right there. There's a nucleus. The nucleus has a membrane around the outside edge. Yeah. Don't remember, it's not the same as an atom nucleus. It's a totally different thing. This is a different kind of situation. And it contains the DNA, the thing that contains our information about who we are. Now, these are much larger. These are from 2 to 1,000 micrometers. Now, how big was a prokaryote? 1 to 10, right? And so this is 2 to 1,000. And so we have a lot larger cells. Hold on, let me get through the slide first. Most eukaryotes are multicellular. So if I say that I'm a prokaryote, it means that I am a creature that is built out of prokaryotic cells. Prokaryote would be built out of prokaryotic cells. A eukaryote is a creature built out of eukaryotic cells. We are eukaryotes because we're built out of lots of eukaryotic cells. Bacteria. Most eukaryotes are multicellular, so we have made of many cells, not just one. Okay. Examples of eukaryotes that are single cellular or unicellular. There's supposed to be another R in there. That's actually supposed to be another L. Sorry. Cellular is what it's supposed to say. Amoebas and some algae and yeast are examples that are unicellular eukaryotes. We will talk about the specifics of the actual organelles later when we get to it. Yes, how large the cell is. Correct. We also have membranes around cells. They can have both a cell wall and a membrane around the cell. No, that's not how we determine. That's not how we determine whether it's eukaryote or not. Eukaryote versus prokaryote is whether the organelles inside have membranes.
Okay, so if it's, so prokaryotic cell can have its own membrane.
A cell membrane, yes, but not organelle membranes.
Okay, I get it.
Okay. Any other questions? Hayden, you with us? Okay. Vivian. Which little symbol? Oh, it's a mu. It's a Greek letter. It means micrometers. So if we talk about 1 meter, right? 1 meter is how long?
3 feet.
Like this, right? About that. That's about a meter. Okay. What is the name we have for 1/100th of that? So the thing we build together again. So yeah. So 0.01 meters is 1 centimeter, right? Now what is 0.001? A millimeter, right? 1 millimeter. This is actually 0.000001. So this is a millionth of a meter. It's a millionth of a meter. It's 5 zeros, which means you divide by 10 6 times. This is the U, yeah, it's a mu. It's called mu. It's a Greek letter. Not like moo. Moo is with an O. It's mu. M-U.
Like meow, it's like meow weird.
Interesting. All right. So those are the types of cells. Eukaryotes. Work with me. Oh. Do not discard. Good. So those are the types of cells. What we're going to do now is we're going to